All right, good morning. So, chapter 7 is about cell membranes. So, the cell membrane ends up being the boundary, right, that separates the living cell from its surroundings, right, the internal environment of the cell from the external environment. And so, what we're going to see is a lot of really important things happening across this cell membrane. If you're going on into microbiology, anatomy, any of those classes, you will think about these concepts a lot in those classes. And so this is a great time to get down some of these fundamentals. And so one of the things I always emphasize to my classes is that the plasma membrane, the cell membrane, is selectively permeable, right? It's going to allow some substances in or out and prevent others from crossing this boundary. And so we'll look at some things cross super easily, like oxygen and carbon dioxide, for example, are lipid soluble. They can just kind of diffuse in and out of the cell. And then a lot of other molecules are going to need some sort of protein to help them get across the membrane. So anyway, lots of fun things to look at here. So as we kind of focus in on the cell membrane, do realize it is a phospholipid bilayer. So what we're looking at here, let me get the pointer. So what they're kind of trying to zoom in here on what a phospholipid looks like. So you have to think back to, was that chapter 5 in the macromolecules? So what we have is we have a glycerol molecule that's attached to a phosphate and two fatty acid tails. So we talked a lot about triglycerides. There's 3 fatty acid tails. In phospholipids, one of those binding sites is now taken by a phosphate. And so what it sets up, this is the more cartoon version here, what it sets up is the phosphate group makes up the head of the molecule. So a phosphate is hydrophilic. It's attracted to water. It gets along just fine. The tails, the fatty acids, remember oils and water don't mix. And so what ends up happening is if you actually just had a bunch of phospholipids and dumped them in water, they basically self-assemble into these little spheres because the tails are so scared of water. They're not really frightened, right? They're just repelled. The tails go together to get away from the water. And so these phospholipid bilayers will actually self-assemble. So again, it's important to know the phosphate end is the head, and it's hydrophilic. It gets along with water, or it's water loving. And then the tail is hydrophobic, water fearing. Yeah, so that's key. Because what this is going to end up doing then is really set up this boundary between outside and inside the cell and what can pass through. The cell membrane, of course, is much more complicated than just a phospholipid bilayer. So this is a model of the cell's plasma membrane. Again, we don't know exactly what it looks like, so this is an artist's rendition, but what we do know is portrayed here. So this is referred to the fluid mosaic model. And in this, what we're saying is we have our phospholipid bilayer and we have proteins. That's these big purple blobs. These are all proteins that are embedded in that membrane. But the reason they call it the fluid mosaic model is the idea is these proteins are just kind of bobbing along in there, that they can move around. They're not, yeah, go with that, right? So they're able to move around. So here's a picture, right? So this is one of the studies that basically fused 2 cells, right? So there's different proteins on the mouse cell and the human cell, but they share the phospholipid, right? So they're very similar. So they got them to fuse, and they noticed that the proteins, right, distributed themselves throughout. So that kind of supported this idea that things aren't set in stone here, but that they move around. And then, so that's the fluid part. The mosaic part is like, if you've seen like a mosaic, right? So a bunch of little broken pieces of tile put together into a picture. Like if you look down on a cell, right, you'd see that. You've got all these little phosphate heads and then just all these little tiles of protein. So that's kind of where they came up with the name. So one thing to keep in mind here, right, is so there's a lot going on in this picture. So let's take a peek here. So we've got some proteins that completely cross the membrane here, right? So that would be referred to as a transmembrane protein. And then, or integral protein is the other name, right, for crossing completely inside and outside are both being touched. And then we have some peripheral proteins. Some of those are on the inside, attaching the membrane to the cytoskeleton. And then there's other peripheral proteins that could actually be holding the cell to what we call the extracellular matrix or the glycocalyx, all these kind of sugars and proteins that coat the outside of our cells. They're also pointing out here, we do have glycolipids. That's a sugar that's attached to a lipid. So we see here our phospholipid has a sugar hanging off of it. And then glycoproteins. So here's a big purple protein with a sugar hanging off the end of it. And those are both going to be really important molecules that we're going to see again as well. So this is kind of your basic picture of the phospholipid bilayer. Oh, check out this one too, cholesterol. So cholesterol is embedded in the cell membrane, and we're actually going to look at how that influences the fluidity of the cell membrane. Okay, so you want the cell to be kind of flexible, right? To have some fluidity. And so depending on whether you used unsaturated fatty acid tails or saturated fatty acid tails, that could make a big difference in how fluid or how viscous, how kind of solid the cell membrane is. So typically that is a difference that something like a bacterial cell would use, right? Deciding whether to use unsaturated or saturated fatty acids in the cell membrane to deal with temperature, right? Because remember, saturated fatty acids are solid at room temperature. Unsaturated fatty acids remain liquid, right? Like oils at room temperature. So the ratio of how you put those together can determine the fluidity of the membrane. That's something that bacteria tend to do. In human cells, in eukaryotic cells, what we're going to see is typically that role of maintaining fluidity is taken over by cholesterol. So at warmer temperatures, so body temperature like 37, cholesterol is able to kind of stiffen up the cell membrane, right? Keep it from just melting. And then at cooler temperatures, it actually maintains fluidity because it's keeping the fatty acid tails on our phospholipids, right, from getting super tightly packed together. And so this is something that we see in animal cells. So when you get into a nutrition class or an anatomy class and people want to kind of make cholesterol out to be the bad guy, you can point out, right, that cholesterol is actually really important in cell membranes, maintaining their fluidity. And they need to have that correct flexibility in order to work properly. Here, so I was mentioning before the different proteins. So this is, I was going to try to get, there we go. It's going to move me out of your way there. So this is a close-up of the protein. So we see our long polypeptide chain here all folded up on itself and folded again. And this is showing, again, a transmembrane protein, right? So it's going all the way through. The other name here, again, integral protein. Okay. There we go. I was like, what did I want to say? Here we go. Let's look at what some of these functions are. Wow, the pointer. Let's look at some of the functions here. So this is huge. Proteins play so many different roles in the cell membrane, right? So what we'll see here is that, for example, we are going to use proteins to transport a lot of the molecules across the cell membrane, right? I said it's selectively permeable, and the phospholipid kind of sets up this situation where really only lipid-soluble molecules can diffuse easily into the cell. But the proteins, right, they can act as channels, they can actually act as pumps. So here we see a protein that's actually using energy, using ATP to force molecules against its concentration gradient, right, basically acting as a pump. We'll see some proteins embedded in cell membranes that are enzymes, right? So here they're trying to show you that it's doing this metabolic reaction. current event with SARS-CoV-2, the protein that it actually attaches to on your cells is an enzyme, right? So the way it docks on your cells and gains entry is by attaching to an enzyme called ACE2, angiotensin converting enzyme 2. Its normal function is actually to convert angiotensin 1 into angiotensin 2. It's a protein that deals with some really important hormones in blood pressure regulation. But anyway, you have enzymes, right? So they're doing these reactions in the cell membrane. We have others that are acting as receptors. So they're kind of keeping this vague here, saying a signaling molecule, just call it a hormone, right? So a chemical, a hormone in your body to get the cell to do what it wants it to do, right? It needs to dock on a receptor and then have that message taken into the cell. The message gets passed on to the nucleus, right, to do something like change gene expression of a cell. It's going to be super important in cell recognition. Right now in microbiology, I'm teaching about the immune system, and this is exactly how something like a T cell, a white blood cell in your body, is going to recognize, say, this foreign coronavirus, right? By checking proteins that are on the surface of cells. We also have some of these proteins that are embedded in the cell membrane that are important in holding neighboring cells together. We'll talk about some specifics of that here at the end of this chapter. But basically, neighboring cells are holding hands, right, with these proteins. In fact, normal cells, if they lose this connection, they'll actually undergo apoptosis. There's something strange. One of the mutations in cancer seems to keep them from listening to this signal. So if they do break free, cancer cells, if they break free from their neighbors, they continue to survive, and a normal cell wouldn't do that. And then this one over here showing, I was mentioning this in the fluid mosaic model picture, right, that some of these proteins are holding on to the extracellular matrix and some attached to the cytoskeleton as well. So tons and tons of different roles that these membrane proteins function in. Mostly today we're going to talk about transport and maybe a little bit of intercellular joining, but these are definitely going to come up again in other classes. So I thought this was a really interesting example that your book gave, so just I thought I'd bring it up. So notice what they're trying to show here are some proteins on the cell surface. So one of them is called CD4, and one of them is this CCR5. So this particular cell, they're not necessarily telling you that, but this particular cell is called a helper T cell, and this is the only cell in our body that HIV, the human immunodeficiency virus, attacks, right, attaches to and is able to gain entry, right? So when the HIV docks on this CCR5 receptor, it's then basically like brought into the cell. It's brought into the cell. So notice in some individuals, and this turns out to be something like 10% of Caucasians, they are missing that CCR5 receptor. HIV can't dock with their helper T cells. It cannot gain entry into their cells. And so they are effectively, I don't even think immune's the right word. Like they just can't get HIV. It cannot get into their cells. So I thought that was kind of interesting. So then that's a target for pharmaceuticals then to say, well, you could think, well, here's a target for genetic modification, right? So that's what the researcher in China did with CRISPR, is he basically took out the gene for that CCR5 protein. But even pharmaceutically, you could think, well, if I could come up with something that blocks CCR5, right, so that HIV can't get to it, then the HIV can't dock and infect cells. So kind of cool. So right, there's like a ton of applications of these proteins. So I did want to encourage you. It's okay to learn this stuff. It might be important at some point. Okay. How do I keep myself out of the way? Okay, so that's the like kind of the basic anatomy of the cell membrane. And so the thing to keep in mind here, right, is form and function always go together, right? And so there's a reason that the cell membrane is set up the way it is, and it's going to very much influence the function. So what we're going to see, again, if we think of the cell membrane as this boundary between inside and outside, this is where exchange is going to take place. So a cell has to exchange materials with its surroundings, right? It needs to dump wastes out of the cell. It needs to bring nutrients and things like that in. And those processes are controlled by the plasma membrane, by the cell membrane. And so it's selectively permeable, and so it is going to regulate that traffic, right? Decide what gets in and out. Now, a couple key things. Non-polar molecules, right? Hydrophobic molecules, things like oxygen, can actually just diffuse right through this membrane. So what we find is, like, water can't just come into the cell, right? It gets along with phosphate, but as soon as water would try to start coming between the fatty acid tails, it's repelled and just pushed right back out. Lipid soluble molecules, so oxygen, carbon dioxide, alcohol, can kind of elbow past the phosphates, and then once they hit the fatty acid tails, they have free access inside of the cell. We'll see the same thing happen in steroid hormones. So like testosterone and estrogen are lipid based. In fact, they're made out of cholesterol, cholesterol is slightly modified to make estrogen and testosterone. And so they just diffuse directly through the cell membrane. Hydrophobic molecules, so things like water, and then also ions, right? Ions have a charge. Other polar molecules don't cross the membrane easily, and those are the ones that we're going to see needing to use these proteins. Okay, and so those proteins are going to end up playing a key role in regulating the transport. So what we'll start looking at is, and I had showed a picture before, some of these proteins called transport proteins, they can act as channels, like they're always open, and they tend to be extremely specific. So it sounds like, oh, there's this open channel, things can move in and out of the cell, but that channel only lets certain molecules or ions use that tunnel, right? So there is a protein that allows water molecules in and out of the cell. Now there's many copies of that protein in the cell membrane, but the only thing going out of, it's called an aquaporin, the only thing going in and out of that channel is water. And then there's different channels for sodium, different channels for chloride, etc. We'll get into a few more of the specifics, because some of the proteins, in order to move things in and out of the cell, they change shape a little bit. And so those are called carrier proteins. They actually like bind to the molecule, and that binding changes their shape just slightly. It doesn't require energy. It's still a passive process. But we've got carrier molecules, and then we'll look at some of the pumps and things like that. So this is really key in understanding how a cell works. And you will see this over and over again if you stick around in biology. So let's make sure we're super solid on this idea of diffusion. And hopefully lab this week will reinforce this. We are going to do a diffusion and osmosis lab because this is one of the concepts I do see students struggle with, and it's really important. It keeps coming back. So what we see here, so we have a membrane. So notice there's some pores, there's some solid sections, but there's some openings. So diffusion at its core is this desire of molecules to move from an area of high concentration to low concentration, right? And so that's what we see right here. If you dropped food coloring in, right, they're showing the actual molecules of dye. Food coloring on one side of the membrane, it's going to, as long as the pores are big enough, that molecule is going to cross the membrane until we reach an equilibrium. And so this equilibrium, the cool thing is, notice some of those molecules are coming back, right? It's free passage both ways, and these molecules just want to spread themselves out. Same thing will happen if you have, here's our yellow dye on one side and our purple dye on the other. So you have two different solutions. They still want to spread themselves out, right? So notice it's not just the concentration of molecules, but it is of those specific molecules. So our equilibrium is reached when there's more or less the same number of purples on both sides and more or less the same number of yellows on both sides. So that's important to understand. So the way this is often described, we say in diffusion, substances move down their concentration gradient. They go from high to low. It doesn't require any energy. There's no work involved here. They just want to spread out. And this is due, probably if you get molecularly, this is due to what's called Brownian motion, that these molecules are constantly moving, and so they're bouncing off each other. And so when they can get further away and bounce off each other less, right, they're happy there. So this is a passive transport. No energy is being expended. Okay, a specific form of diffusion kind of governs how water is moved around. So osmosis. Osmosis is the diffusion of water, right? So the movement from high concentration of water to low concentration water. Osmosis is the diffusion of water across a selectively permeable membrane. And so what usually confuses students about this, right? So what they've set up here is they have this selectively permeable membrane. It's selectively permeable in that the pores in this membrane are small enough to let water through, but not small enough to let this solute through. I think in this case, this is like a big molecule, like a sugar or something. So water can move freely across here. So notice on our both sides of our little membrane here, there's way more sugar, right? More solute on this side than there is this side, which means, and here's where students get confused, look at the close-up, there's way more water molecules on the side that has less sugar. And so water still wants to move from an area of high water concentration to low water concentration. And so it wants to go across the membrane and join this side. And so see how the water has actually pushed across the membrane and the whole column rises. And now we reach equilibrium, right, where we end up with more or less the same concentration. So water is crossing this selectively permeable membrane in osmosis. One way that I find that students like to think about it that seems to help is this idea that water follows salt, right? Or water follows the solute. So technically it's going from an area of high water concentration to an area of lower water concentration, but that area of lower water concentration, it's got a lower concentration because there's more solute there. And so the water is following and going to that side. And so again, if this helps you to look at this close up, right, the water molecules can pass through, but the sugars can't. And so this side has fewer solute, more free water. And so now we see the majority of the arrows going towards the right. That's osmosis. Let's play with it in lab. Make sure you understand it. Here is why it matters, right? So the rules of osmosis, I'm just getting out of the way. The rules of osmosis are important because it affects, right, our living cells. So there's a thing, there's got to be a better word. There's I cannot come up with a better word. Tonicity. So tonicity is the ability of a solution, right, surrounding a cell to cause that cell to gain or lose water. Let me say that again. Tonicity. It's the ability of a surrounding solution to cause a cell to gain or lose water. So tonicity is a term that is always a comparison, right? We're comparing the solution outside of the cell to the solution inside, right? The cytoplasm. So the tonicity of a solution depends on its concentration of solutes relative to that inside of the cell. And so what we're seeing here in this picture are three different descriptions of the fluid that our cells are sitting in. So in an isotonic solution, the solute concentration is the same inside the cell and outside of the cell. So as water is moving in and out, notice, right, water is moving in, water is moving out, but there's no net water movement, right? And so the cell is just like normal, right? No water gained, no water lost, because the saltiness is the same on both sides. It doesn't pull the water in a direction. Now, if we look here on the right, this is describing a hypertonic solution. That means the solution that this cell is sitting in has more solutes in the solution, right, outside of the cell, than there were salts in the cytoplasm of the cell. And so that means, right, if the outside is salty, that the cytoplasm has a higher concentration of water, and water is going to move out, right? And so we see water moving out of these cells, and these cells are shriveling, right? Technical terms like crenation, in a plant cell, it's plasmolyzed. Okay, now, a hypotonic solution works the other way. The solute concentration is less than that inside of the cell. So this is like distilled water or something, right? High concentration of water outside of the cell compared to inside, right? Because it's an extremely dilute solution. That means water is going to want to move into the cell. The cell gains water. Now, in animal cells, that is actually going to cause the cell to explode. In plant cells and fungal cells and bacterial cells, that water can actually come into the cell and kind of expand and push, right? So it can expand and push, but it has a cell wall that prevents the cell from actually rupturing. And so, in fact, you'll notice that this is actually when the plant cell is the happiest, right? So it can fill, remember, the central vacuole from last chapter, that water can fill the vacuole, right? And the water pressure pushes then out on the cell wall, and that cell, they call it turgid, right? It's full. This is normal for a plant cell. And notice in an isotonic solution, it can't actually pull enough water into the cell, and so your plant is kind of right? Leaning over. Okay, so this ends up being super important. You'll see this over and over if you're continuing on in a medical field. I mean, think about it. If someone gets an IV, do you want their IV to be hypotonic, isotonic, or hypertonic? You want an isotonic IV. And so in humans, we give a 0.09% NaCl saline. That matches the salt levels inside of our cells, and so all our cells are happy to receive those fluids. Okay, and so then this is just another example of how life deals with osmosis. And so this is a paramecium, a little single-celled protist. And so what these guys have done is they have what's called a contractile vacuole. So particularly for single-celled organisms, hypertonic and hypotonic environments can create real problems. And we would call those an osmotic problem, right? It's under osmotic stress, right? Which way is the water going to go? Because just like us, Paramecium doesn't have a cell wall. And so these organisms need to do what's called osmoregulation. They need to control solute concentrations and water balance, right? This is a necessary adaptation. And what we'll see is that organisms that live in fresh water might do this a little differently than organisms that do this in salt water. And so what the Paramecium has done to solve this is it has a little what's called a contractile vacuole. Remember, a vacuole is just that little like membrane bound sac inside of the cell. And they actually use this as a pump. So if the cell takes in too much water, they put it in the vacuole and then they can dump it back out. So pretty clever way to get away with living in Paramecium live in fresh water. And so water would constantly be rushing into the cell, risking lysis or rupture. And so they're going to use this vacuole as a pump. So kind of a cool real life example. Okay, so these are kind of a couple major ways. I just keep being in the way here. A couple major ways this works. So when we look at these proteins, some of them act as channels. And I was talking about this earlier. A channel protein is always open. Now notice they're talking here, they're calling this facilitated diffusion. Anytime you see diffusion, you should realize that this is a passive process. No energy is taking part in these reactions, right? And so these channels are players in facilitated diffusion, right? They have the correct channel to allow a certain molecule, ion, etc. through. So again, these are extremely specific. Some only let water through, some only let sodium through, some only, right, let chloride through. We will see some really cool examples of some of these channels. So I say they're always open. There are channels that are called gated channels. They actually open and close in response to a stimulus. So you'll see this in anatomy class. You'll have these voltage gated sodium channels. So it's actually like this is how neurons send messages, right? How nerve cells send messages. A little wave of electricity actually triggers the opening of a channel and lets sodium into the cell. So there are places where these are gated, right? But still totally a passive process. The carrier protein, this is the one that always frustrates students. They're like, I don't understand how this is passive. So what you have to think of, right, this carrier protein, right, it's open to the, say, the outside of the membrane at this point. When that molecule binds, remember this is a protein, and so the shape of the protein is very much dictated based off of bonding and intermolecular reactions. So when that molecule binds in the carrier protein, that's actually changing the shape of it slightly. It doesn't require any energy, right? But it does open in the other direction. This is also always going to be diffusion. It's always going to be moving molecules down its concentration gradient. So this is a passive process, no ATP involved. Now we do have times when we want to use ATP, and this is the classic example. This is the sodium-potassium pump. So anytime, so those other processes were passive, this is considered active transport. Active transport requires energy, and typically this is going to be in the form of ATP, because that's the cellular currency of energy. So this is, for example, this is a carrier protein, right? You notice that it changes shape, but this one is actually using ATP, and it is going to be able to move things against their concentration gradient, right? So that's a key thing there as well. So what we see in the sodium-potassium pump, right, is that the way cells work best is if outside of the cell you have high levels of sodium and low levels of potassium, and inside of the cell, it's reversed. You have high levels of potassium and low levels of sodium. So what ends up happening, though, is this sodium-potassium pump is what sets that up, what allows that to happen, right? Because what happens is sodium is always trying to leak into the cell and potassium is always trying to leak out of the cell. And the sodium-potassium pump just keeps bailing them back in the direction that the cell wants them. So here we see sodium binding, and then by using ATP, we shove sodium out of the cell. It was already a high concentration of sodium out of the cell, and we're going to force it against that concentration gradient and add more. And then notice while we're open, two potassiums can move in. And this side is actually, this is a passive process, right? When potassium binds, the protein changes shape again and dumps potassium into the cell against its concentration gradient, right? There's already a lot of potassium inside, and we're just adding more, right? And so again, sodium is getting kicked out of the cell and potassium is being brought into the cell. I left those zoomed in versions in case you need to read through that more closely. Okay, and there are other pumps, but that's kind of the classic example. Okay, and so again, just to review, some molecules, lipid-soluble molecules, diffuse directly through the cell membrane. We have channels that allow facilitated diffusion. We also have carrier proteins that allow facilitated diffusion. And then active transport, we see this carrier protein requiring ATP to serve as the energy for moving those ions. All right, we can check this video out in class. Here's another one that we'll see, and this will actually come up here in a couple chapters when we start talking about energy. This particular protein is acting as a proton pump. So it's moving a hydrogen ion, which is just a proton, across a membrane against its concentration gradient. And we'll see that this requires ATP. This can set up some neat opportunities, right? There's a lot more going on than I'm able to talk about in this chapter. What this is trying to show is what's called cotransport. So what happens here is, notice there in this particular protein, we're looking at the diffusion of hydrogen down its concentration gradient. And the way this particular cotransporter works is that as hydrogen is trying to move down its concentration gradient, it actually makes it so that sucrose can kind of sneak a ride through. Notice that sucrose is actually moving against its concentration gradient, but you don't see any ATP over here, right? So it's like hitching a ride for free inside of the cell. Now, this whole transporter is only working, though, because we have a proton pump down here using ATP to force all the hydrogens against their concentration gradient outside of the cell. So they want to come rushing in, and they'll go ahead and bring sucrose with them. So our body's life has amazing ways of getting these things done. And so one of the things to keep in mind is the way that cells move solutes in and out of the cell is super important because it does then determine whether water is attracted, right, and wants to move in or out of a cell. So again, cotransport, just to make sure we're clear, cotransport is occurs when active transport of one solute, that's our proton, indirectly drives the transport of other substances, right? So that's our sucrose kind of sneaking a ride in. So pretty cool. Okay, other active ways that we can move molecules in and out of cell. This is what's called endocytosis. So in endocytosis, there's a few different forms here, right? So they're showing us phagocytosis, which is more like cell eating, pinocytosis, cell drinking, and then receptor-mediated endocytosis. And this is where receptors on the cell membrane are quite specific for what we're trying to bring in. So what endocytosis is for is the small molecules, ions, water, things like that. We were seeing them leaving or moving into the cell across those transport proteins. But larger molecules, so that big old chunk of food there, like a big polysaccharide or a protein or something, can't, it's too big to get through a transport protein. And so we're going to move it in bulk in a vesicle, right? And so that's what endocytosis is. It's this bulk transport that does require energy. So in endocytosis, cells are taking in macromolecules by forming vesicles in the plasma membrane. And those are our three kinds. Cell eating, and that's, I thought that was super cool, right? Here's, look at this amoeba trying to eat that algae, right? It's gonna have to kind of have to keep trying, right? That algae is pretty big. Pinocytosis, again, drinking. This is, I think, a really fascinating example. So this is a way that a cell is able to ensure that it's getting very specific solutes, right? So the receptors only fit certain molecules, and then when enough of them have been filled, have been attached to, then we'll go ahead and create that transport vesicle. Basically, once you're in this vesicle, right, we can pull those little purple triangles off and then put those emptied receptors right back into the plasma membrane. And so an example, again, just trying to, I know some of this stuff is like, it can seem really boring, but it's super important. And so there's an example in humans, right? We use receptor-mediated endocytosis for our cells to take in cholesterol, right? So again, we were saying cholesterol is important in cell membranes. So in our bodies, we have cholesterol molecules called LDLs, right? Low-density lipoproteins. That's who delivers cholesterol to our cells. And then we need receptors. Excuse me. We need receptors, these purple guys, in our cell membranes to attach to LDLs and bring them into the cell. And so there's a disease called familial hypercholesteremia. And so that should tell you it's inherited, right? Familial. Hypercholesteremia refers to the fact that you're going to end up with high cholesterol in your blood. And that's because people who have this disease don't have receptors for LDLs, or they don't have enough receptors for LDLs. And so those cholesterol molecules aren't getting taken into the cell. they just end up staying in the bloodstream. So a real life example for you. Anyway, there we go. That is it for chapter 7. Again, lots of super important stuff in there. So do take the time to check that out. And we'll see you soon.